Welcome to the Wednesday afternoon lecture series. I'm John O'Shea and NIAMS, and I'm really thrilled to introduce today's WALS speaker and William Paul Award speaker, Yasmin Belkaid, an NIH alumna and a longtime friend who's made remarkable advances that you're going to hear all about. But before we start with the introductions, I have to make two very quick announcements. One is the first is the code for CME credits. The code is 66834, and I have to repeat that again, 66834. And also for those who are watching remotely, you can submit questions to the speaker anytime during the lecture. Simply click on the button that says Send Live Feedback, which is below your videocast window, and type in your message, and we'll relay it to the speaker at the end of the lecture. And next, what we'll do is we'll have Ron Germain, who's going to tell you a bit about Bill Paul, since Yasmin is the Bill Paul speaker. And so why don't I let you take it away, Ron?
And it's really a pleasure and a privilege to have the opportunity to remind all of you, or for those who don't know, tell you something about Bill Paul, for whom this lecture is named. And as you can see in the slide, Bill is the immunologist. Now, this was his professional photo, but we remember Bill much more this way. He was always an ebullient, engaged, smiling person who never was happier than discussing science. So a little bit of background. Bill recalled in one of his memoirs how he became an immunologist, or at least decided he might become an immunologist. He was reading a small volume by Michael Heidelberger, the immunochemist, on a trolley car in Brooklyn during his college days, and he says he was smitten. He then deviated, I have to say, and became an endocrinologist for a couple of years at NIH, but then realized his mistake, went back to immunology as a postdoctoral fellow with Baruch Ben-Asaref at NYU, and then moved with Baruch when Baruch became the chief of the Laboratory of Immunology at NIAID. In 1970, Baruch moved to Harvard, and Bill was named the chief of the laboratory, that time the youngest chief of the laboratory here, and he served as LI chief for 45 years. Now, he's made way too many scientific contributions for me to name them all, but let me list a few. He discovered BSF1, B-cell stimulatory factor 1, which eventually was cloned and renamed IL-4, a very famous cytokine. He was the principal person who dissected the role of IL-4 in IgE responses, how the receptor signaled, and the rules governing Th2 development and IL-4 and 13 production. He was an essential player in the development of an absolutely essential concept in adaptive immunology, which is the role of positive cytokine feedback in controlling the flavor or subset of Th development. He also helped uncover the roles of different cytokines in regulating the isotypes of immunoglobulins that were made by B cells, matching the T cell production of cytokines to the effector function of the antibodies that were made. And then, kind of as a sidelight, he spent years with Ziv Grossman working on theories of how self-tolerance is maintained. And I really can't take the time to tell you many of his other discoveries, but he was a much bigger figure in the field than that might show you. While he was here at NIH, he helped establish the NIH Vaccine Research Center through proposals that went up through Bill Clinton and Al Gore together with Tony Fauci. And he also served as the first director of the Office of AIDS Research. In the larger community, he was on, I've seen his CD, and there are pages of the advisory panels that he has served on. He helped guide selection of honorific awards, such as the Lasker, advised academic institutions, and also disease-focused funding organizations like the Lupus Foundation, which has named an award after him as well for his service. And then he was an amazing educator outside of his mentoring role that's already been discussed. And through his textbook, Fundamental Immunology, and his action as the editor of the Annual Review of Immunology for nearly 30 years. And during the period he was the editor, The annual review of immunology was the most highly cited biomedical publication in the world. He also was a terrific mentor. He's received the AI Mentoring Award, and just a few names you might recognize: Charlie Janeway, Ron Schwartz, Laurie Glimsher, Mark Davis, Bob Cedar here, Jeff Zhu, and Josh Milner. And let me end by saying that Bill had a special sauce as a scientist. Most of us help manufacture small threads, but they have to be woven together into a tapestry to really understand the patterns in the natural world. And there are only a few people who collect those threads and actually weave the tapestry that allows us to understand how biology really works. And Bill was such a weaver. He had an enormous intellect, a remarkable focus, and tremendous experimental rigor. And he was able to pull all of these things that both his lab developed and other people developed into our current concept of adaptive immunity. And so let me end again with this photo that to me represents the Bill I always remember. And so, John, back to you.
Thanks, Ron. Before I introduced Yasmin, I'd just like to point out that I was an adopted member of Bill's lab at the time with Mark Davis's cloning the T cell receptor, Lloyd Glimcher's work, and MHC and autoreactive T cells, and as Ron just said, the discovery of IL-4. And I have two vignettes that sort of I'm thinking about Menschlichkeit and clarity. And what you're seeing here on this slide is at that time when Bill was thinking about cytokine feedback and subsets and the like, and we found that interferon gamma induces TBET, the master regulator of Th1 cells, and we submitted our paper. And it managed to get it sort of be rejected by all of the big paper, the journals and everything, and it worked its way down. And I went to Bill and was, crying on his shoulder and saying, what can we do? And Bill was, despite the fact that it was rejected multiple times, Bill said, no, this is really great. This is really terrific. We can help you with this. And Bill said, actually, well, we'll put it in PNAS, don't worry about it. And needless to say, I said, no, this is really great that you're helping me with this. And it was just that idea of being of real mentorship now this paper is cited many times. Next slide there, Diane or Chris. The other thing I'm very proud of course, is this collaboration with Bill Paul and sort of anticipating the idea of the plasticity and of helper T cells that really preceded single cell RNA-seq, which is now commonplace now. But I want to go to the next slide here, which I was trying to articulate a concept, and I was being, I guess, very, in my mind, thinking very creative. And I had this idea about these master regulators looking like smoky mountains, and I sort of generated this. And this cartoon is still actually in PubMed. But as Bill and I were talking about this and going to the clarity point, that Bill said, it is really lovely, John, but I think no one's going to understand what you're talking about. And so I said, well, how would you put it, Bill? Next slide. And then he just put this in this very simple way of thinking about that all of these subsets, they really had, gradations of these different transcription factors. And it was just really great. And this is where the final version of this cartoon was published. All right, so again, I'm very grateful for learning so much and working as a collaborator with Bill. And I'll stop there and start introducing Yasmin. And it's going to take me, she has so many great things to talk about. It'll only take another half an hour or so, Yasmin, so don't worry. So Yasmin was born in Algiers and began her undergraduate and graduate training, including the Pasteur Institute of Algeria, working on leishmania. And then she earned her doctorate in immunology from the Pasteur Institute in 1996, where she continued to study innate immune responses in Leishmania. Came to the NIH as a postdoc in NIAID, Laboratory of Parasitic Disease. And she did really landmark work in David Sax's lab back when FOXP3 positive cells were brand new. And Yasmin identified that Tregs control Leishmania. In 2002, she joined the faculty of the Cincinnati Children's Hospital, but returned to NIAID in 2005 as the head of the mucosal immunology unit, in LPD, and went on to have multiple leadership positions at NIH, lab chief of laboratory of host immunity and microbiome, director of the NIH Center for Immunology, director of NIAID microbiome program, and now has been appointed president of the Pasteur Institute from January of 2024. Again, lots and lots of things to say. She was on the editorial boards of Immunity, Cell Science, Journal of Experimental Medicine, numerous meetings that she organized over the years, major discoveries from TGF-beta and retinoic acid induction in FOXP3, the importance of GATA3 and FOXP3 Tregs, pathogenic Th17 cells, series of papers, control of skin immunity by residential commensals, and including recent publication related to antibody response to commensals, mate cells imprinted by microbiota, and most recently, mammary intraepithelial lymphocytes, how they promote lactogenesis and offspring fitness. And again, too many awards. I'll do my best to get through it quickly. Gold medal award from the International Union of Biochemistry and Molecular Biology, Sanofi Pasteur International Award, Phillips Foundation Award, the FNH Lurie Prize, Von Buring Prize, Koch Prize, Arab Genius Award. I like that one in particular, the Genius Award bit. The AAI Thermo Fisher Meritorious Career Award, AAI Excellence in Mentoring, which again, I think everyone's very proud of that kind of award. She was elected to the National Academy of Sciences, National Academy of Medicine, American Academy of Arts and Sciences, Académie des Sciences, Chevalier de la Légion d'Honneur, and the UK Royal Society. So I'll stop it there for Yasmin and hear about all your great stuff. Thanks.
Hi, everybody. Can you hear me? Yeah?
Yep, can hear you.
Well, thank you so much for this introduction, John. And it's an immense privilege for me to be invited to this lecture. And I really, truly apologize for not doing that in person. I mean, I would have wanted to be here and just be back at the NIH, and in particular, to honor Bill. Because as it was meant before, Bill was the immunologist of the immunologist. And for me, he was an icon, and he was a role model, and he was someone that really inspired my science for many, many years. And I'm going to try to actually, along the way, during this lecture, honor him through, in particular, a story I wanted to talk, and about the leadership he had actually done in actually promoting and protecting the development of vaccines to the VRC. So I'm just going to very briefly summarize what has been said earlier, which is actually I started my life in Algeria. And starting my life in Algeria also was an inspiration for the rest of my scientific journey, which is very much exposure to a world where infectious disease were still there, infectious disease that still affected people and were still affecting people, in particular parasites. And I also remember stories from my childhood of my own grandmother having lost children with infectious disease. And for me, working on host-microbe interaction was very much inspired by where I came from, and really became a conductive line across my career. So I started, as it was mentioned before, in Pasteur Institute in Algiers in parasitology, but it is really at the NIH that I became an immunologist. And I really had the extraordinary privilege to spend pretty much all my scientific career as an independent investigator within the NIH, where I was really inspired by an extraordinary community of scientists coming from all around the planet, working on immunology, infectious disease, vaccine development, and so many more. All of them had a major role in my own scientific career. So along the way, during the last 30 years of my scientific life, I have really worked on many aspects of host-microbe interaction. And really, us and many others have really realized the extraordinary complexity of those relationships that can actually go all the way from alliances, as I will discuss further in the context of the microbiota, for example, all the way to microbial threats. And the middle of this is really the immune system that is really constantly dialoguing with these microbes and tailoring the ability of this microbe to either coexist or become infectious and actually dangerous for the host. And as I mentioned before, a lot of what I have been doing and the immunologist I became at the NIH was very much inspired by the community of the NIH and this extraordinary leadership that immunology had within the intramural campus, very much led by the formidable figure of Bill. This community of the NIH was also the one that had generated the IAG, the Immunology Interest Group, that was generated by at the NIH, and really also was an environment where we could actually work together and collaborate. So this really strong link between immunology and infectious disease also played a major role in my next step in my career, which is really within the Institut Pasteur. And just wanted to actually spend 1 minute to tell you about the Institut Pasteur and what it is. So the Institut Pasteur is actually a private non-profit institution that is essentially focused on infectious disease, but not only. It was founded by Louis Pasteur 130 years ago. It is highly transdisciplinary. But what is really extraordinary about this place is not only the fact that as the NIH, we are highly international, it's 110 nationality, but also the fact that Pasteur is connected to 33 institutes across the planet, allowing to work in a highly collaborative environment on diseases that affect the world. So very much the experience I had within NIAID in particular, an environment where we work on infectious disease in a global manner, was really one of the inspiration for me to take this new position. But what Pasteur Institute is also is the birthplace of what can be considered modern vaccination. And this will actually relate to the rest of my presentation. Vaccines are and remain the safest and most effective tool of public health to allow protection. And this is actually how we harness our immune system to actually protect. I'm showing you here on the left, an image of Jenner, first vaccination in 1796. And of course, talking about Pasteur, the very well-known rabies vaccine that he had developed and actually really shared with the world. We know that actually vaccines have actually changed and curved the trajectory of infectious disease. We knew that recently in the context of COVID-19, but we know also that child mortality has been actually profoundly impacted. And then actually now almost one child on 2 was dying before the age of 5. And today we, of course, have actually dramatically changed this worldwide. I'm just showing you this slide to actually illustrate how one single disease, measles, was really responsible of most of child mortality. And actually, this is actually the result of the last 50 years, showing actually how many children have been saved just by the measles vaccine alone. So just all of that to actually remind you of the importance of vaccines, something that, of course, had been a strong focus of the VRC and one in which actually Bill had played a major role.
Dr. Belkaid, I'm sorry to interrupt, but your slides are not showing.
Oh, really?
Yeah.
None of them? Okay.
No, and I'm sorry I didn't see that earlier on Teams.
Okay.
But the
so
video cast are not showing on the video cast.
Okay. So how do you want me to how do you want me to share that? Because I just used the team and I shared. Is there any other link I could use? Do you see them now?
Something is coming up now as we speak, yeah.
You see that?
Yes, I'm sorry I was in the wrong page view. You got it now. I apologize.
Okay.
Looks good.
Okay. Okay,
Yep.
good. Okay. So I just wanted to actually remind you once again the importance of vaccine worldwide, but also the fact that infectious disease remain an important threat for humanity, and we're going to need new vaccines and new approaches to develop them, because many of the pathogens that actually exist do not have existing vaccine. And this is of extreme importance because we know that not only we have emerging pathogens, this is caused in large part because of change in the environment, deforestation, displacement of population, but also we have re-emerging infection. And more importantly, we also have a fast rise in antimicrobial resistance. And we're going to need to actually be able to develop at a high pace new form of vaccine and new strategy for vaccine against pathogens that so far would have no way to protect our population. So the question I'm going to ask during this lecture is the following one. Can we actually learn from our evolutionary partner to develop new vaccines? And when I refer to evolutionary partners, I'm referring to the microbiota. So we're very lucky when I came back to the NIH after having had a lab at Cincinnati to be given the opportunity by NIID to develop a program on the microbiome. And this is really the power of the intramural, the ability of allowing people to take risk at a time in which this field was really just emerging. So I was very lucky to benefit from this resource that allowed me to develop a program that allows us to be among the first to actually discover the fundamental role of the microbiome in the control of the immune system. And what we're able to show is that the microbiota was able to control the ability of the host to develop response to infection, was able to amplify and shape the quality of vaccine, and it was actually subsequently shown by lipulendrum that actually antibiotic treatment could actually change the glycosylation pattern of antibodies in the context of vaccination. It was also shown that the ability of the microbiota to push immunity could actually have an impact on immunotherapy, on immune checkpoint therapy. And this was actually also nicely, beautifully shown by Giorgio Trinchieri, who actually showed remarkably that you could take the microbiota of responders to immune checkpoint therapy in the context of melanoma and transfer them to non-responders and restore some of the responses. And this actually is becoming the basis of a new understanding of harnessing the microbiota as an adjuvant, but also the ability of the microbiota to control the immune system can have negative impact in the context of autoimmunity, inflammatory disorders, and allergy. What actually became also intriguing in laboratory, besides the fact that the microbiota was this extraordinary adjuvant allowing the development and function of the immune system, was actually another phenomenon that was of high interest to us. And it was the following one. Having read, with attention as an emerging immunologist textbook, and having actually been influenced by, of course, Polly Matzinger and Genoa, of the importance of actually damage and inflammation to initiate immune responses, I was really left, as much as many others, with a conundrum. If really there is a need for inflammation and damage to trigger immune responses, how come the microbiota that is covering every surface of our body is able to actually engage the immune system without causing inflammation? Indeed, we know that the microbiota not only control the immune system, but that the immune system recognizes the microbiota, and you can generate both B cells and T cells against these microbes. So this really led to a series of questions over the last few years, and in particular to try to understand what could be the function and the mechanism by which these actually immune responses directed to the microbiota are engaged. And I'm going to here summarize some of the work, but first, of course, give credit first to a truly remarkable graduate student in my laboratory who actually started to collaborate when she came with Julie Segre and Heidi Kong, which of course both at the NIH, that were really the pioneer in exploring the skin microbiota in humans. And what Shruti did was really to try to address the following question: could the skin microbiota, in a similar manner that what we have shown in the gut, control local immunity? And she was able to show experimentally that the skin microbiota was indeed able to control local immunity in a manner that was able to influence immunity to pathogen. So just a couple of words about the nature of the skin microbiota. They are actually not floating around at the surface of the skin. They are mostly embedded strongly within infrastructure such as the sweat glands, the hair follicle sebaceous gland, and the biomass of this microbe is very low. So these data were actually quite fascinating because it meant that this very low level of microbe is sufficient to shape the local function and the local immunity of the tissue. So what Shweli, but also others in laboratory subsequently showed is that when the microbiota colonize the tissue, these microbes are actually thin. And what I'm not going to describe with you today is actually then this microbe are actually able to engage in a quite remarkable way with the endogenous retroviruses, allowing really a form of very small signal, allowing the induction of these immune responses. T cells generated against the microbiota migrate back within the tissue where they become tissue resident, and within these sites, they influence local physiology by enhancing antimicrobial defenses. They can also be highly plastic, coming back to the point that John had mentioned, change the phenotype and engage in epithelial repair. They can also induce neuronal regeneration. This was a recent work of Michel, via the ability of the cells to produce IL-17, and can also even promote a broad antitumor immunity milieu. So this class of immunity was different from the one that actually we had explored in the context of pathogenic interaction of vaccine. It was a class of immunity that was induced in absence of classical inflammation, both in terms of induction and when the cells went back to the tissue. So this actually led us to explore the following question. This was very much, inspired by Inta. Inta was also a remarkable, and is still a remarkable scientist that came in my laboratory as a postdoctoral fellow, and since then has started her own laboratory at London University. And I forgot to mention that Shruti is now really a rising star at Mount Sinai, really as an associate professor. So the question that Inta had was, besides T-cell responses, could the skin microbiota be able to induce antibody responses? So to address this question, Inta applied the bacteria at the surface of the skin of an animal in a very simple experiment. These animals are not germ-free, and she was able to show that just having those bacteria at the surface of the skin is sufficient to induce a significant generation of antibody against the bacteria, IgG1, IgG2b, IgG3. And this actually happened in absence of any form of scratching, any form of actually abrasion. This is just a topical application of a microbe at the surface of the skin. This phenomenon of having antibodies against the skin microbiota is not a mouse phenomenon. This was actually in the context of a collaboration with Janet Busben and Michael Fischbach, and I will come back on this collaboration later. When you actually survey the serum of individuals, healthy individuals, you can find a very high level of antibody, circulating antibody against a large array of skin microbes. So of course, you could speculate that this is caused by maybe abrasion of the skin or in the context of infection, or we could also speculate that just the presence of those bacteria at the surface of the skin may be sufficient also in humans to induce antibody responses. But if this is the case, how does it happen? So the first question we had was to try to see what could be the role of the skin commensals and why would you actually generate so much antibody against this microbe. And of course, the first hypothesis, which is not surprising, will be that this antibody could serve the purpose of actually protecting the host against the possibility of this microbe becoming pathogenic. Indeed, the microbes are never fixed within one single form of interaction. And even Staphylococcus epidermidis, that is, of course, a skin commensals, can become a pathogen in the context of, for example, a medical device, or in the context of a child that is born actually prematured. So having antibody against the microbiota could, of course, serve the purpose of preventing infection in this context. So to address this question, what Inta did is the following question. She took animals and infected them intravenously with Staphylococcus epidermidis. In this context, this is actually can be seen as a pathogen, and the bacteria can be found within the spleen, for example. If she now had applied the same bacteria at the surface of the skin a few weeks before, then challenged the animal with a pathogen, the animal is actually protected, vaccinated against this pathogen, as you can see here. Importantly, this is entirely dependent on B cells, because if you utilize animals that actually do not have B cells or animals that could not produce antibody, this protection is abolished, really pointing to the fact that this antibody preemptively can actually avoid subsequent infection, and that a topical application of the microbe is a form of vaccination. So to try to understand the class of immunity generated, INTA utilized animals that were deficient in AID, that is a master regulator of somatic hypermutation and class switching recombination, and showed that if you do not have AID, the protection that was afforded by topical association is abolished, and also, of course, then plasma cells are going to be important for this response. So this actually really shows then this response is highly specific and really depend on the maturation of a highly classical form of immune responses and highly matured antibody. So the field and numerous people in the field, Pam Schwarzberg that is here and many others, have actually decoded the extraordinary complexity of how you generate high-affinity antibodies. And this actually has been shown to be occurring within specialized structured germinal centers in which you have coordination of responses between B cells, T cells, TFH, allowing the evolution of these responses and production of antibody. So to try to see if this topical association could generate in a similar manner than in the context of a vaccination or infection, those germinal center, INTA, of course, looked in the regional lymph node connected to the skin and was able to detect the presence of those germinal center. But what was quite surprising is when she started to look within the skin itself. She was able to see that within the skin, you could also see those germinal center-like structures that express a plethora of molecules and cells that resemble classical germinal center that have been detected and characterized extensively in the lymph node. So to try to understand if those structures in the skin were akin to really what happened in the lymph node, she utilized animals that were deficient in molecules known to contribute to germinal center reaction, and in particular IL-21, really central molecules of this response, CD40 ligand, and as well as BCL6 that will, of course, eliminate the possibility of TFH, a T cell responsible for this response. And you can see that in the wild type, you have the generation of those germinal center-like structure, or TLOs, but you can see that this is not the case in animals that were deficient in the other factors, really pointing that the molecules responsible for generation of those local responses are the same than those responsible for the germinal center in the lymph node. So, of course, the next question that INTA had was to try to see what could be the nature of the antigen-presenting cells responsible for this response. And when she observed the presence of these cells in tissue, she could see then, for the most part, these structures were really surrounding the hair follicle that is really the natural home of the macrobiota. This is really where those microbes are actually living. And she was seeing an enrichment, of course, of lymphocytes, but also of dendritic cells within this environment. So numerous scientists in the field have actually decoded the complexity of the dendritic cells network in the skin. And I'm going to highlight here one population in particular, that is Langerhans cells, that is in fact more macrophages than the Langerhans cells, and is in fact the first cell I worked with when I was an undergraduate, trying to really understand the function of these cells. And I have to say that for the most part, despite the fundamental density of these cells in the tissue, Langerhans cells are probably the most abundant population of antigen-presenting cells in the body, their function remained largely unclear. When she looked at this hair follicle, and you can actually find an accumulation of Langerhans positive cells that were really present within the hair follicle, but also of a chemokine that is extremely important for the generation of germinal centers. And this, of course, was the first hint that the Langerhans cells could be important in this response. But the real proof of the role of these cells came from the utilization of an animal that allows the deletion of Langerhans cells using a diphtheria toxin injection and exclusively remove Langerhans cells, but not other population of dendritic cells, as you can see here. And using these mice, INTA was able to show that in absence of Langerhans cells, IgG1, IgG2b, and 3 are all abolished in absence of those dendritic cells, really pointing to a non-redundant role of Langerhans cells in this response, and really demonstrating that maybe one of the main functions of this extremely dominant population of cells at the in the epidermis is really to control the macrobiota and the relation with the macrobiota. But the next question that INTA had is the following one, which is, she had demonstrated this production of TLOs in the outer layer of the skin. And the question was to try to understand if this could have really a role in the generation of antibody locally. And the reason this was of potential interest is because that hadn't been really explored before. An antibody, especially in those homeostatic conditions, are going to be believed to be really generated within within actual lymphoid structures. So this is just to look at the BCR frequency within the skin, and you can really see that in the skin, she identified an enrichment in clones that could actually suggest an enrichment and antigen-specific responses, and not something that could be just a bystander response. And importantly, when she looked within the skin itself and she looked at those TLO, she could also identify AID within the skin, really pointing to the idea that maybe there is a local activity of this enzyme in the tissue, showing the possibility of this response to be generated locally. But to address this response, or to address this question in a more precise way, what INTA did is the following experiment. She utilized animals that were lymphotoxin deficient, which means they have no secondary lymphoid organ, and remove actually the spleen to make sure that they had absolutely none present in the animal. Then applied wild-type animals, or animals that have absolutely no secondary lymphoid structures, with staph epi, and looked at antibody generation. On the bottom, you can see here the lymphotoxin alpha deficient mice that are splenectomized can still generate TLOs. And this really points to the fact that these responses are not secondary to something that will have happened in lymphoid organ. This can actually happen in absence of any secondary lymphoid organ. When now she looked at antibody responses, you can see that IgG1 is completely abolished, really pointing to the fact that IgG1 depends on lymphoid structures. But if you look at IgG2b or 2c, these responses are largely maintained. So this really points to the fact that certain isotypes are actually maintained and can be produced in the skin independently of any secondary lymphoid structures. And it was actually the first demonstration that the skin may have an autonomous ability to generate antibodies. To further look at this point, she really looked at IgG2b expression within an animal that have no secondary lymphoid organ because they're lymphotoxin splenectomized. And you can see here that you can detect a very high number of IgG2b expressing cells in these animals within the skin itself. To go a bit further and actually to see how far those responses go, she utilized a very simple experiment in which this time she swabbed the animal that had been applied with staph epi and looked at antibody-specific responses at the surface of the skin. And you can see here that using this approach, quite simple, she can detect IgG to be specific from staph epi just by a gentle swab of the skin, showing that those antibodies are produced in such a high amount they can actually exude and be present at the surface of the skin. So more recent work that is actually unpublished, that is actually led by INTA in collaboration with Sinisa, that is actually a collaborators that has a huge expertise in glycosylation of antibodies, highlighted something really, really intriguing. What INTA and Sinisa have actually done is really compare the glycosylation pattern of antibody that can be generated within the skin or in the blood. And what they can actually show using actually chromatographic approaches is a striking differences in the glycosylation pattern of antibody produced in the skin as opposed to those that are expressed in the serum, really pointing to the fact that where the antibodies are going to be generated is going to have a major influence on glycosylation pattern that could really influence the function of this antibody. And this is something that we are now currently exploring and to try to understand the functional relevance of this striking observation. But the next question, of course, was to try to understand the role. Why would you generate antibody locally? We know that the microbiota, as I mentioned before, exists in the continuum, all the way from commensalism and actually a symbiotic relationship, all the way to pathogenic infection. But even as a symbiont, the microbiota has to be kept at bay. This is what we have learned from Andrew McPherson's work and IgA in the gut that really help the microbiota to be maintained away from the tissue and prevent those partners to become invasive. So the question that INTA had is, could these local antibody responses serve the important purpose of maintaining the skin microbiota in its symbiotic form? To address this question, what INTA did is went again to a very simple approach, apply the bacteria at the surface of the skin, took an animal that was wild type that will of course have antibody both in the skin and systemically. And what she actually did is utilize a gentle swab to really look at the biomass of the microbe that remain in the skin post-association. As we have shown before, the microbes that are present will be present in very low biomass. If she utilized an animal that have no antibody responses, here animal that will be deficient in TFH, these animals have no antibody in the skin or in a systemic manner. And you can see that now the microbes that are applied to the surface of the skin really are in a very high biodensity, which means that actually now the skin microbiota becomes like a weed and really proliferate at a massive level, something that is clearly not a desirable situation, as those microbes can potentially now become a source of infection. So clearly antibody not only serve the purpose of preventing subsequent potential infection, but serve also the purpose of maintaining the local biomass of these microbes. But now how about the local production? Here we have an animal that has no secondary lymphoid structure, and you can see that those animals can only have antibody at the surface of the skin. And you can see that only antibody produced by the skin are absolutely sufficient to control the biomass of microbe, pointing to the fact that this local production of antibodies serve the fundamental purpose of maintaining healthy relationship with our skin symbionts. So you can see here, and this is actually summarized on the right, then the CD40 ligand knockout mice, this is actually exactly the same experiment that I have shown here. An animal that have no antibody responses, the skin microbiota become really overrepresented. But if you have animals that have that have only antibody at the surface of the skin, this response is actually constrained, really pointing to the fact that these local antibody responses is physiologically relevant and important. So the model that we actually have built based on this observation is the following one. INTA has actually uncovered a new role for Langerhans cells, which was the first antigen-presenting cells discovered, and that is actually to constrain and to allow the ability of the response to the microbiota. And this was actually done, of course, by Steinman, the discovery of Langerhans cells. You can actually see that these Langerhans cells can coordinate two different responses. One that is actually classical in the lymph node, allowing the generation of IgG1 that serve the important purpose of preventing subsequent infection with the skin microbiota, and one that is actually local, that is allowing the generation of different isotype. And I think that by itself is quite remarkable, that you will generate different isotype based on localization. And more importantly, these isotypes are differently glycosylated within the skin as opposed to other tissues. So this actually opens many, many different questions. Why would you generate different isotopes? What is the function of those? How do they diffuse in the tissue? What do they bind to? And is a glycosylation pattern play any role in the ability of this antibody to act locally? I haven't actually showed this data because actually this is published, and if you're interested, feel free to look at it. But we found that the ability of this TLO to be generated and TFH responses locally relies on the conversion of regulatory T cells locally toward the TFH phenotype, maybe providing one explanation of why you have so many Tregs within the skin environment at any given time. So we believe that this actually compartmentalized immunity to the microbiota allow to provide two things. One, of course, the protection against subsequent infection. The second, the coexistence, the peaceful coexistence of the microbes. But this actually, of course, opens a new doors of question. I'm going to come back to the beginning of my lecture about vaccine. Is can we utilize that as an inspiration for design of vaccines? And especially, as we all know, the fundamental importance and challenge of vaccine is to reach antibody levels within tissue that will be sufficient, and also to generate enough antibody that will be present at mucosal sites. So this is actually where our long-term collaboration with Michael Fischbach came in place. Janet Busben had a very parallel observation in ORS about antibody generation in the microbiota, and we joined forces in a publication that really tried to actually look at how much this observation could lead to design of new vaccine. And this is actually what the experiment that was done by Janet, in which she took Staphylococcus epidermidis, same microbe that we have utilized, and utilized an enzymatic conjugation, which is actually it's actually a transient a conjugation, which actually is extremely important because it will allow potentially to use that in the clinic. This is not a genetic manipulation of the microbe. She applied the bacteria at the surface of the skin, and quite remarkably, she was able to generate antibodies against the bacteria, but also against any antigen she could actually couple to the bacteria. So it's quite remarkable that just taking an antigen and actually coupling in an enzymatic manner to Staph epi allows the generation of high titers of antibody just by topical application of the microbes. So what was also quite interesting when she looked at it is that was actually also able to generate a very high level of nasal IgA antibody. And we all know the importance of generating IgA within tissues, showing to the fact that actually this strategy seems to have an important role to actually generate this response. I'm sorry, I haven't updated that, but it was actually published in Nature this year. But the most striking experiment they have actually done, which actually is a proof of principle that this could be a vaccine, is the one that is shown here, is when they actually label the bacteria and they utilize a tetanus toxin antigen. And they utilize a lethal dose that allows the mortality of some of the animal. If the animal had been applied with the bacteria that was actually coupled transiently with the tetanus toxin antigen, the animals were entirely protected against these lethal challenges. This actually demonstrates really that the topical association of the microbe expressing foreign antigen in a transient label manner is absolutely sufficient to provide long-lived protective immunity against this antigen, and in the context of this highly lethal challenge to protect. So of course, this is just the beginning of a story, and we believe that much more needs to be done. And we have now currently a collaboration with the Michael Fischbach with the Gates Foundation to try to push this further. But I think there is a lot to learn from evolutionary partners, and in particular in the context of vaccine design, how to maybe actually utilize the way by which those microbes have co-evolved with us by really taking advantage of this relationship with the microbes and our immune system to actually design a new class of vaccine that could allow us to generate a class of vaccination that allows high level of titers within tissues, and in that without causing any inflammatory disorders. So I just wanted to remind you that infectious disease is not a history of the past. They remain a major threat to humanity, and actually continuing to work on vaccines, developing new vaccine against these emerging and re-emerging threats, developing vaccine against antimicrobial resistance pathogen remains an important task that we have all to collectively continue to engage. On that, I have to thank all the remarkable people that have done the work. A special thank to Inta. She's a remarkable scientist that's going to have a huge impact on the field. She's now started her own laboratory. It's impossible for me on this slide to say thank you to all the people that I am grateful to at the NIH. NIH and IID has been my home. I have more friends there than I can actually list. And I just wanted to tell you how grateful I am for all what you have done, all you're doing, and for all what you're representing. You are an extraordinary group of scientists. And I just wanted to thank you all for having been such incredible friends, mentor, having actually been here for my trainees. Special thanks to all of you. Just here a picture of my laboratory, the last picture of my laboratory before it closed, but also of my new emerging laboratory at the Pasteur Institute. I will just try very hard to remain a scientist despite my slightly busy schedule. This is too important today to remain a scientist. And I just wanted to take a minute to tell you that if any of you that is watching this is interested, Pasteur Institute is recruiting junior faculties, and do not hesitate to actually send your application. It's an extraordinary environment, and we are looking forward to receive many of you here. And on that, I just wanted to thank you all and to take any questions if there are any.
Thank you. Clapping here. There are a few questions, Dr. Belkaid. I'll just go down in the order they came. Fantastic talk. Would you please comment on how the skin and hair products, such as cream, hair dye, may affect the functions of the immune system on our skin or hair follicles?
think it's a very interesting question that is not addressed. I think we are an animal outside of our ecological niche. How much what we do in our skin is changing the response we have to microbes is an interesting question. I mean, we're using harsher product. We have a study that was done recently by Alex Wells in a laboratory that shows that certain kind of detergent can actually change the physiology of our skin. So we do not know actually in the context of this specific question, but it's a worth question to explore. How much local antibiotic treatment that we have, how much actually the change in the pH, change the nature of this microbe, and as such, the response we have against them. It is clear that we have to make sure that our relationship with microbe is maintained in a peaceful way, because this microbe, as much as they are positive, can become also a source of inflammatory disorders.
Okay, thanks. Is there any indication that the presence of skin microbiota and their ability to stimulate the skin immune system has any relevant effect on vector-borne pathogens' ability to infect the skin and produce pathology?
Well, this actually is interesting because that brings me back to a long time ago where I was transiently a medical entomologist. Absolutely. I mean, think about having, first, it's known that the skin microbiota influence the attractiveness of mosquitoes. That is very interesting by itself. If we have a different kind of immune responses in the skin that is influenced by the skin microbiota, this will change also the nature and the inflammatory nature of the milieu in which the pathogen is going to be transmitted that could have an influence on the pathogen. So I think it's a very, very interesting question that hasn't been formally explored, but I think it will be extremely important to actually integrate that in our understanding of the first milieu in which the vector is transmitting the pathogen. This milieu is going to be conditioned by all these existing factors, the microbiota, its ability to control the immune system, the recall of these T cells to the microbiota that is now going to enter the tissue. So I think this is a very interesting question that will need to be integrated in our understanding of vector-borne disease.
Okay. I think you get this next one a lot. Does the chronic use of antibacterial soap have any impact on these immune responses?
You know, probably. I mean, we're not the one that's going to express that, but it's, you know, the microbiota is not going to just disappear every time you take a shower. So we should remain completely confident in our ability to. Nonetheless, I think harsh products are clearly not a good idea. I just would like to remind us that we are an animal and we actually have co-evolved with microbes for a reason. So I think anything that does strip away the diversity and the healthy relationship with these microbes is not an innocent act on our physiology. All animals remain colonized by the microbiota and have symbiotic relationship with them. So when we actually act in a way that is too aggressive with those surfaces within the skin, the lung, the GI tract, we're changing the nature of this relationship, and this has consequences.
Okay. The letters ABS, is that antibodies perhaps? Because
yes.
The question is, are there ABS? Are there antibodies against T-cell independent antigens of bacteria such as polysaccharides?
Oh, that's very interesting. Yes, I'm sure there will be some. We have not explored that. The one we have looked at were the ones that are classically maturated and but this is a very good question. Is there any natural antibody that I suspect there could be? We haven't really looked at that, but that's an interesting question.
Okay, one more before I have to process some other ones. You showed nasal IgA. I wonder whether gut IgA also follows the same trend and whether it is possible to create sustainable, quote unquote, antigen-specific gut IgA titers in the gut.
So IgA responses can be generated by all microbiota of the gut. That has been this actually the first well-characterized immune response to the microbiota was really well-characterized in the gut. So you could imagine that if you absorbed orally some skin microbe from various reasons, you will generate IgA responses. What was really remarkable in the work of Janet is how easy it is for her to generate IgA responses by this topical association of microbes. She can put them in the nasal layer, but she can also put them in the skin and can generate that. So there is really something about the ability to generate high titer IgA responses via these approaches that is quite unique. And she's now she's applying now to independent position and is going to be exploring that in her own laboratory.
Okay, Dr. Germain, you want to jump in?
Yeah, Yasmin, it's really spectacular as always. But one thing I may have missed, and I know we talked to Inte when you were here, the germinal center responses that typically occur in secondary lymphoid tissues nucleate in the follicular dendritic cell network. Do you actually see FDCs in these GC in the skin, or is there a substitute stromal structure?
Yeah, we don't see FDCs, so I think you're absolutely correct, and there will be something else that is actually stepping in. So she see a lot of the factors, chemokines, anything that could actually be linked to those responses, but does not see those FDCs. So clearly something else is stepping in to take stage. And she's now, this is exactly what Inta is trying to decode using, spatial transcriptomic and others to really try to really understand really more how this is really coordinated. But this is not FDCs, no.
Thanks.
Okay, I have a few more. What are the differences?
I have to say that
please,
when Ron actually asks a question and say it starts with "but," I'm always scared.
What are the differences between adults and children about the skin immune system?
That's a very, very good question. I think in terms of all immune system, we're not as knowledgeable as we should have been, especially when it comes to immunity within tissue. So it's a very interesting question. In terms of the skin microbiota, for example, we know that there is a dramatic change in the skin microbiota in puberty, for example, but we don't really know enough about how the child is actually developing a relationship with the skin microbe early in life. We don't really know in general about how the immune system develops in each tissue. And this is extremely important if we want to actually develop strategies to actually manipulate the immune system for vaccine response and others at different age. So in terms of the microbiota and response to microbiota between a child and an adult, I'm not sure I can actually add much more than that in terms of the skin. I'm sorry.
Okay. This questioner may have a patent in mind. Can we manipulate Tregs to regrow hair?
Oh, I'm sure that many people will be happy. I don't know if we can do that yet. We also know that actually adaptive immunity and aggressive immunity in the context of the hair follicle is one of the factors that actually lead to the destruction of the hair follicle. So, maybe Tregs could be a strategy to actually improve the retention of hair. That's an interesting question, and we haven't tried that, but maybe an interesting question. In any case, any form of way by which you can actually prevent aggressive responses within the hair follicle and destruction of the hair follicle, part of it could be mediated by the immune system, is a way to potentially just prevent all of that. And, an interesting question, for example, is how much aberrant immunity to the microbiota contribute to the further destruction of the hair follicle is not something that is really known.
Okay. Is it clear how much bacterial load is necessary to induce these immune responses? For example, if you are exposed to a new microbe in passing, such as a handshake, is that enough exposure to induce a response?
So I don't have this answer, but I have another one. So Inta did something, really clever experiment. She took animals that she applied with the bacteria, and then she co-housed them with another. So basically, the animal acquired the bacteria passively in the same cage, and they were able to generate antibodies. So I don't know if it's a handshake. I don't know what the mice are doing. But the acquisition passively of the microbes in the same cage was sufficient to trigger the response. I think work that was done years ago showed that actually you can actually acquire microbes by changing environment. For example, it was shown classically that changing hotel room can actually allows you to acquire new microbes. How much of those could be coupled with acquisition of antibody responses, I think will have to be experimentally tested, but in human.
Okay, Dr. Jumay, maybe you can ask the final question, and then maybe either you or Dr. O'Shea can wrap things up and passing comments or final words.
Yeah, well, let me broaden the question. One of the things you mentioned along the way was the role of endogenous retroviruses in driving the responses to the commensals. Mm-hmm. There are lots of people on long-term antiretroviral therapy now. Have you, or is anybody exploring what happens to the local immune system in the skin in individuals who have the suppression? You used, antiretrovirals to show that was actually the mechanisms. We know that they should have this effect if you can extrapolate from the mice. So
Yeah.
what's happening with these people who are on long-term art therapy?
You know, it's very interesting what you said because we started I was at this meeting, a dermatology meeting recently, and it was really interesting to talk with dermatologists and their experience as clinicians with patients that are on long-term retroviral therapy. So we're now starting to look into that, which I think hasn't been looked enough. We know that those treatments are extremely important. They're saving life and they're remarkable. Yet they have actually, as all treatment in the world, have some consequences. But there is clearly skin phenotype. There's clearly exacerbation of certain inflammatory disorders. And we are now actually in the context of looking into that more precisely. What was actually shown, the only tissue in which it has been well explored is in the vaginal compartment, where it has been shown and actually you can actually have a diminution of the immune system and the intensity of the immune response in the tissue. I think the jury is still out and we're going to need to do the work. But it's interesting to talk with the clinicians that are actually following the patients and the observation they have actually generated. So hopefully in a couple of years, I could tell you more, John.
Great. John, floor is yours.
Oh, wow. That was just amazing, Yasmin, as always. And it's just such a great, way to honor Bill. I could just imagine him sitting there in the front and be asking these questions and just be so excited about the work that you're doing. And you're a terrific person for this award. And thanks for joining us. We would have liked to have had you here. You were busy doing important stuff, but we will need to get you back here soon. Thanks,
Thank you very much,
Yasmin.
John. Thank you so much for everything. And I don't think there's anything more important than science. So thank you all, really.
All right.